So what I'm going to, I've got kind of a jam-packed talk for you today, so try to pay attention, okay? Because, and the title is in a common model of information spaces in human cortex. And so common means that it's a model that of what is shared across brains in terms of how information is represented and processed and how connectivity between areas is organized. And information spaces, I used to say representational spaces. Now I'm saying information spaces because of our recent work in which we have extended this common model to the human connectome. So this is just the current and some emerita people in the lab. Some of you will recognize him already, like Jarek, who's now a research professor here. Fei Long is a current graduate student, and Sam Nastase is a current graduate student. And Swaroop Guntapalli just left his postdoc after he was a graduate student here and then a postdoc for a little while. And now he's working in private industry at a company called Vicarious. So this is something that upsets me. He should be, he should be staying in academic science because he's one of the most extraordinary people I've ever worked with. And losing him to private industry hurts. And then I just have to acknowledge also some people at Princeton in electrical engineering that we've been collaborating with for many years. And we've burned through a sequence of graduate students, starting with Rory, with Murtsabunku, who's now at MGH, and the current one, Cameron Chen, I think he just got a job with Google. So this is an outline of what I'd like to talk about today. First, I'm going to state the problem, which is how do we capture both coarse and fine-grained topographies in human cortex in a common model, meaning a model where we find bases that are shared across brains. What will these bases look like? And we'll start by talking about the conceptual framework we use is high-dimensional representational spaces. So we start off with voxel spaces, but we project that into there should be high-dimensional information spaces. And I hope that will make sense to you after I talk about it for a while. And then I'll talk about how we derive this common space in individual transformation matrices using a method called hyperalignment. So we can take individual brain data and derive a transformation matrix that projects that subject's brain space into this common model space, or project data from the common model space back into an individual subject's anatomy, which can be that subject's own data or data from other subjects. And a bunch of validation tests, and we're going to talk about how this can be applied to connectivity in the human connectome. And then I'm going to talk a little bit about how in this model we model topography, and we model it as multiplex or overlapping topographic basis functions. And this is relevant because Sam Gersman presented another way of doing that yesterday, but our method is really quite different from his. Then I'm going to have conclusions, but I'm not going to be done if I have any time left over, because I want to talk a little bit about how we can now start to look at individual differences in the framework of this common space. So I don't know if I'll get through all this or not. So let's start off with the basics of decoding. And that is that we can look at the pattern of activity that's evoked during an experiment and try to decode what the experimental condition was that evoked that pattern of activity. Now we're talking about patterns of activity. Everything I'll be talking to you about today is done with fMRI, but the math and the methods also applies to MEG, EEG, and single unit recording. So this is a study that Andy Connolly did and published in 2012, in which he had subjects look at still images of 6 different species of animal, with 2 species of insect, 2 species of bird, and 2 species of primate. And in the decoding analysis, we use a support vector machine, SVM, to try to classify the patterns of activity by the stimulus. So this is a confusion matrix over here. And the stimulus that was viewed is here along the vertical axis, and the classified output is along the horizontal axis. And so a good confusion matrix has nice bright colors along this diagonal, which means that The patterns evoked by ladybugs are classified as ladybug patterns. The patterns evoked by warblers are warbler patterns. And you can see this looks great. This worked really well, and we can classify all these animals, including find distinctions of one bug versus another, or even one primate versus another. This is a lemur, ring-tailed lemur, and this is a squirrel monkey. And the surprising thing is that many of our subjects didn't even know the difference between lemurs and monkeys. So, but their brain made a distinction between them. Now the problem is that this kind of a classification analysis is always done in individual subjects. So we take one subject's brain data and we divide it into training data where we build the classifier model, and then we test it on the left out test data to see if it's valid. And, what that means is that for each subject we have a different classifier model. Now if we take the classifier models from some subjects and apply them to a new subject, where the data have been just aligned in terms of anatomical features, it just doesn't work very well. So this shows the result of that. This is the average classification performance for within subject pattern classification with a mean accuracy of about 68, 69%. This is the confusion matrix if we do a between subject classification, building the classifier in 11 subjects data and then testing that classifier on the left out 12th subject. And you can see it's not as pretty, and the classifier performance crashes from 69% down to 37%. So whatever is used by the within subject classification analysis is not shared across brains. And what we contend is that that's fine scale structure within these brain areas. This is in ventral temporal cortex. Now, how do people think about the topographic organization of the brain? Now, classically and currently, most labs think about the organization of the brain by dividing the brain into brain areas. This has a long history. This is the Brodmann map from 1909, in which Brodmann divided the cortex into 52 areas. People still refer to brain areas when they present their experimental results. More modern methods, like that of Evan Gordon, working with Steve Peterson and Laumann, et cetera, have used resting state connectivity, and they divide the brain here into 422 areas, which they say is fairly reliable across subjects. So now the problem is that this implies that a brain area, like the precuneus or lateral occipital cortex, is a homogeneous brain area. Okay, it's an area and it does one thing, and there's no information in this kind of an aerial model about what is the structure within these brain areas. But we know there is structure within our brain areas, and that's very well described in early visual cortex, and we know it exists in later cortical, not strictly sensory cortical areas also. So this just shows the organization of early visual cortex, and V1 is the first yellow area here. This is from the work by Marty Serino. And then V2, and this is defined by retinotopy. So the fundus of the calcarine sulcus has a representation of the horizontal meridian, and the border between V1 and V2 is where the representation is of the vertical meridian in the retinotopic field. So there is a structure within V1 with a single cycle of polar angle and a single cycle of eccentricity. And there are other layers of structure within V1. So this is work from Yakub from the University of Minnesota, and showing that you can image the ocular dominance columns. This is all inside V1, okay? And you can divide it between columns, hypercolumns that take input from the right eye and columns that take input from the left eye, shown in red and blue. And there's a yet finer level of topography, functional topography, with a map of orientation selectivity. So this is from the same section, and actually, if you're sitting close to the front, you can see black outlines here, which are the outlines of the ocular dominance columns. And within each ocular dominance columns, all orientations are represented in these orientation hypercolumns. So we know in V1 there are multiplexed topographies for retina topical location, ocular dominance, orientation selectivity, also for color and motion. Now what about after we get out of early sensory cortex, where we understand the topography and can label these different multiplexed basis functions, what about things like ventral temporal cortex? Well, ventral temporal cortex is usually modeled in terms of areas. These are that are category selective. So this is Nancy Canwisher's 2010 paper in PNAS, showing the centers for areas that are place selective, face selective, body selective, and word selective. And this is a more modern take on it by Colonique Grill-Spector, showing the location of the face areas in red. There's an occipital face area and a posterior and a middle fusiform face area in red. But again, the structure within these areas is not addressed by this kind of model. But we know there has to be fine-grained structure in FFA. There's lots of evidence about fine-grained structure. So from our fMRI studies, we've been able to decode what species of animal, of mammalian animal face is the subject looking at, a human face or a dog face or a monkey face, even though they all evoke strong responses in the FFA. From the patterns of activity, we can decode if the face one is looking at is a full profile, three-quarter profile, or full face. Is it one individual or another individual with the same angle of profile? Is it a monkey or a lemur? And even, is it a shoe or a chair? All these kinds of information can be decoded from patterns of activity in the FFA. So there clearly is structure in the FFA, fine-grained structure, that is accessible at the spatial resolution of fMRI, but we don't understand what the basis functions are for that. We just know that it exists and it can be exploited for pattern classification and other types of multivariate methods like looking at representational geometry. So the question is, can these fine-grained topographies that carry fine distinctions be modeled in a computational framework that is common across brains? So it's very easy to do it in V1 with known basis functions for orientation selectivity and retinotopy and things like that. It's a little harder when we get into areas that are higher level, like ventral temporal cortex or even prefrontal cortex. So to tackle this problem, and we've been working at it for a long time now, trying to build a common model. Swaroop started at graduate school here in 2008, and I put him on this project immediately, so it's now 2017. And we're not done yet. In fact, we have lots of mysteries that we're still trying to solve. But we think about representation or information in the brain in terms of an information space, okay, rather than a cortical sheet. And so projecting data from a cortical sheet into this information space is very simple. So this would be the pattern of response to, let's say, monkeys, warblers, and Luna moths. And each one of these is a row in a data matrix. Each column in that data matrix is a voxel. And so you can think about this vector response, pattern vector response for monkeys as a location in a high-dimensional space in which each dimension is a voxel. I'm showing a two-dimensional space here. This is a 10-dimensional matrix. The real matrices we work with tend to have 200 to 50,000 dimensions. So now each one of these experimental conditions, stimulus categories, is a vector in an information space. Now, the same framework works for functional connectivity. So instead of stimuli, if you look at connectivity targets, so here's a connectivity target in prefrontal cortex, parietal cortex, anterior temporal cortex, and each one of these connectivity targets, connectivity to voxels in a brain region, is going to be a little different. There's a topography there, and you can conceptualize that topography of connectivity for a connectivity target as a connectivity profile vector. Before I talked about a connectivity pattern vector. Before I was talking about response pattern vectors. Now we have connectivity pattern vectors, but it's the same math. It's a data matrix. It's got rows that are vectors, and these rows that are vectors live in an information space with the same dimensions as the dimensions in the response space. So we're talking about the same space. We're just talking about two different kinds of information in that space. Okay? So, and this implies that there's a fine scale structure in the human connectome. And I'm going to show you that that's true, something that's been unappreciated before. So how do we do this? How do we take brain data from brains of different sizes where things don't line up very well and think about them in terms of an information space? That's what these things are supposed to indicate. This is the same data as the brain in an information space. What's the kind of transformation? That's the third column here. That's going to put them into a common space. So you can think of it this way. You've got, we have people watch a movie. We have them watch Indiana Jones, Raiders of the Lost Ark from beginning to end. And so at this point, the evil archaeologist Belloc has just stolen the golden idol from Indiana Jones. Everybody who sees this is kind of experiencing the same thing in the movie. They hear the same voices, they're seeing the same scene. So in lateral occipital cortex, these three voxel patterns represent pretty much the same information in the movie. But because they're poorly aligned, even though these may be the same anatomical loci, the patterns look different. So in their individual representational spaces, voxel spaces, the vectors of these three patterns are in different locations. That's what these red circles are here. Later on, Indiana Jones escapes in a seaplane. And so when people are seeing the seaplane take off, they have patterns of activity in lateral occipital cortex, and those now, the green vectors, are in different locations, but again, they're not lined up across subjects. So somehow, we want to get these vectors, so vector number, this green vector for these green vectors, to be in the same location in a common space. So somehow we have to calculate transformation matrices that will put them in a common space so that now the red vectors, that's the evil archaeologist Belloc, and the green vectors, that's the seaplane escape, are in the same location. So how do we do that? Is this possible? And there's an assumption here that if we have one transformation, vectors, response pattern vectors, from a wide variety of experimental conditions are all going to line up with one transformation matrix, which is a pretty outrageous thing to assume. But we're really leveraging the fact that we know that representational geometry is highly correlated across subjects, meaning that the shape of the pattern vectors, the distances between pattern vectors for different stimuli is very consistent across subjects. And that's why what we're leveraging here. So how do we do this? Well, we have people watch Indiana Jones. So we tip our hats to Steven Spielberg. And so here's an early scene. And this is just to let you know that we are not conventional psychologists. This is not a controlled experiment. It's very cluttered, it's dynamic, there's lots of information. This is key, though, to this kind of building common models of brain spaces. You need a very rich data set to have a very rich sampling of response pattern vectors. I would contend that the resting state human connectome data is also equally rich in terms of getting a very rich set of connectivity patterns. Now, these are the responses that were evoked in two real subjects while they watched the movie, and these are in sync. And you can see that they don't look much like each other, even though we're assuming that this information represented here is very similar to the information represented here. It's not obvious how they're similar. And that just shows that when we look at things anatomically aligned, we don't see the similarity. So this is another way to think about it. So these are the same two subjects. And as they watch the movie with each new pattern, you can think of that as a vector in the representational space of each subject. So you can see these vectors appearing with each new response pattern. And they're color-coded, so the blue circles are the same time point. The green triangles are the same time point. And they really don't line up, right? So how can we get them to align? Well, there are a couple ways of doing this. We use a a rigid rotation. So it preserves the representational geometry. It's an improper rotation for those of you who are a little geeky about this because we allow reflections. But if you, we use something called the Procrustes transformation, which calculates the optimum improper rotation to align two sets of vectors, of labeled vectors. And it's a closed form solution. It's not an estimation kind of procedure. It just calculates the optimum. Yeah?
So how you calculated the response pattern vector? Is that corresponding to each response pattern vector corresponding to each scene of your movie?
Yeah, each time point of the movie, each time point, so each TR, okay, is a pattern. And we just look at, that's what we use. There's a lot, and if you want to talk to me later about all the grisly details about normalization, et cetera, but that's basically what it is. So it calculates a rotation, and watch carefully. Oops. Okay. So we want to align this pattern, this cloud of vectors, with this cloud of vectors. So we have to calculate this transformation matrix, which we do the Procrustes transformation, and it rotates these data, and now the vectors have the same pattern, okay? And if you project them into, if you overlay them, now the vectors for the same time point are really close to each other. This, of course, is illustrative data. A third subject now, has a different pattern of vectors for the same time points in the movie. So the transformation for subject 2 doesn't work for subject 3. You have to calculate a new transformation matrix for subject 3 that will now rotate that subject's data to be in alignment with the mean vectors of the first two subjects. Yeah?
So do you need to assume that voxels never like stretch or contract between subjects in order to do this? So maybe I overrepresent my meridian.
Yeah, so that's really a good question. And what we're doing is we're resampling the information in voxels into these model dimensions here. And so it's a weighted sum. And so the information for one voxel can be spread out over a number of voxels. But in terms of, is it like a functional area, expanded or contracted? There's some of that, but we don't model that explicitly. Okay. So, and then there's we have to aggregate the transformation matrices for local cortical fields into a matrix for the whole brain. And we constrain the resampling of information from a voxel to a dimension, which is referenced to this first subject we call the reference subject. So it's all referenced to the first subject's anatomy, but it's not identical to that. So we aggregate this so we get a whole brain transformation matrix. With this whole brain transformation matrix, now we can take this subject's brain space and project it into the common model space, and take any data in the common model space and project it back into the subject's anatomy using the transpose of the transformation matrix. So this is the basic equation, and that is that we are deriving a common model. This is for a cortical field J. So this is data in the common model is the mean of the individual subject's data for that cortical field, B sub I, times subject-specific transformation matrices, which are rotations, R. Okay? So that's what the model data in the model space is the mean of subject data, of transformed subject data. And this is done to minimize the difference between each subject's transformed data and the model data. And that's why we use the Procrustes transformation. Now, with our collaborators at Princeton, we've tried a couple of other algorithms, like regularized canonical correlation analysis, which may work a little bit better than Procrustes. Probabilistic hyperalignment, this is Cameron Chen's work. He calls it now the shared response model. And this seems to work quite well, but we can't figure out how to take the data from the probabilistic hyperalignment and aggregate it into a whole brain matrix yet. But as I said, this is work in progress, and this is going to keep me busy and happy for a long time to come. So does this work? So this is the idea, but it seems kind of far-fetched. Oh, shoot. Okay, for some reason, I have this problem. So a row in the brain voxel space, okay, is a pattern of response across voxels for one time point. And after multiplication with the transformation matrix, I've got to figure out why this doesn't work. That pattern for that response in that subject's brain space is now a pattern of responses in the common model space, where now the coordinate axes are the model dimensions, not the individual subject voxels. So the goal is so that these patterns of response are now more similar across subjects, which should afford better between subject classification. So to test that, we classified the movie time points themselves. So we asked, can we say when from the brain data that we collect while subjects watch this movie, what part of the movie are they watching? So we take 15 seconds of brain data, that's 5 TRs, and ask 5 or 6 TRs, depending on the data set, and ask, is the pattern of activity in that short segment of time most similar to other subjects' activity during the same period of time as compared to all other times during the movie? So when they see the seaplane escape, do they all have the same pattern of activity that's different from the other things in the movie, like the Balak, or the scenes in Egypt, or the scenes with the submarine, or the scenes in Nepal, right? I don't know if all of you, I've got this movie memorized. I've seen it so many times. But when we do that kind of a classification analysis, this is a correlation-based nearest neighbor classification analysis. On the anatomically aligned data, the only areas that show classification performance above 5% is back in primary visual cortex. Now, we use a sliding time window for alternative time segments, which would be errors. So the chance classification is less than 1 out of 1,000. It's about 1 out of 1,300. Okay. After we do the hyperalignment, we can now classify with 5 to actually about 45% accuracy in all these places in the brain. So primary visual cortex, lateral occipital cortex, ventral temporal cortex, prefrontal cortex, parietal cortex. It's just a huge increase. As a matter of fact, if we look at this quantitatively, so you can see the numbers in areas that are identified using neurosynth, for visual areas in red and auditory areas in blue and cognitive areas in green, you see a huge increase in classification accuracy in all these areas, in occipital, temporal, parietal, and prefrontal cortices. That's the blue bars as compared to the red bars. And overall, the classification accuracy in these different functionally defined loci is about 7 times higher. So we increase local between subject multivariate pattern classification 7-fold. And if we do this for the whole brain, and the way Swarup did this is really Swarup's work. The way Swarup did a whole brain analysis is he did a singular value decomposition and looked at classification using different numbers of singular vectors. And after hyperalignment, it peaks somewhere around 300 to 400 singular vectors. So it seems to be about the dimensionality of this information space that we're harvesting that's common across subjects. And the classification accuracy is 93%. So that means with 93% accuracy, we can say exactly what part of the movie that someone's watching at any time. Pretty high. Anatomical alignment was surprisingly high at 76%, a lot lower than hyperaligned, and it peaked with a dimensionality of about 50 dimensions. So we get a much higher dimensional information space with much finer grained information by using hyperalignment. I'm sorry about the missing numbers in here. So a column in the subject's brain space is the tuning profile for a voxel. And we model, we get a tuning profile for a model dimension as a weighted sum of the tuning profiles for voxels. That's just how the linear math works. And so if we look at, are those tuning profiles more correlated across subjects, this is the correlation of local tuning profiles down below after anatomical alignment. After hyperalignment above, you can see it's a dramatic increase, nearly twofold. Then we looked at representational geometry. And again, but I'm going to talk more about this, so I have to explain it. With the movie, we can also look at representational geometry. Usually, in the representational geometry papers you've read, people will have 5 or 10, or with Krieger's cord to maybe 94 conditions, right? We have 1,300 conditions because we have 1,300 time points in the movie. And we looked at the correlation between the response at one time point and the response at all other time points. So you can imagine if Indiana Jones is in the scene, it will be correlated with other scenes with Indiana Jones if those are areas that encode faces, right? Or if there is a certain theme music at one point, other time points that have the same theme music will be more correlated, right? So you can look at this kind of geometry for all the information that's in a movie. And if we look at whether or not the geometry, local geometry in searchlights is correlated across subjects, it is correlated across subject after anatomical alignment, but the correlation, again, doubles after hyperalignment. So we're doing a much better job of finding the common local representational geometry with this. And again, we do this for these 20 regions of interest. And you can see we get increased representational geometry everywhere. Now, we had a long haul getting this paper published. And one of the nasty criticisms that we had to deal with is this is just the same thing over and over again. There are global features in the movie that are driving this representational geometry. It's not specific to the information represented in different cortical fields. So we had to address that. So to do that, we looked at a second order representational geometry, which means we looked at, is the representational geometry in one of these cortical fields, like MT, how correlated is that with the representational geometry in other visual fields or in auditory fields or cognitive fields? So we're looking at the correlation of the representational geometries across brain areas. And this is the matrix, dissimilarity matrix of representational geometries for these 20 cortical fields. And it's hard to digest, but you can see V1, which is this red bar over here, is different from everything, maximal dissimilarity. The visual areas are most similar to each other, the auditory areas are most similar to each other, and these cognitive areas have kind of a complex pattern where the calculations are more similar to visual areas, and this working memory on the right is more similar to the auditory area for some reason. But you can visualize this kind of a dissimilarity matrix using multidimensional scaling. So we project all this into a three-dimensional multidimensional scaling space. And that's what's shown here. And again, this is nicely color-coded with vision in red, audition in blue, and cognition in green. And this is the first dimension, the second dimension, this is the first dimension again, and the third dimension is the vertical axis on the right. And you can see that the first dimension is dividing vision from audition with cognition somewhere in the middle, and the third dimension is dividing cognition from the sensory perceptual areas. And within vision, everything's bunched up together because this is a solution that's optimizing the structure for all 20 areas. If we simplify this and look at just what's the structure within those visual areas, so we did another multidimensional scaling, two-dimensional scaling, and we found that the first dimension divides V1 from everything else, and the second dimension divides these higher order areas with MT down here, word form, face area, and the place area on the top. And in the auditory domain, the first dimension, again, is primary auditory A1 versus the higher level auditory voices and music. And then dimension 2 seems to be dividing right hemisphere representation from left hemisphere representation. So in other words, we're getting very meaningful structure in the second order representational geometry that indicates that the information that's being represented in these different fields is different. And it's related to, we think, what they're supposed to be representing, like auditory information and visual information. Now, we've gone on with this and used it in other areas. So this is I hope Matteo doesn't object, but I'm showing one of his figures. So here's Matteo, and he's working with Ida Gobini, and they've analyzed, this is the same movie data, with different areas in the face processing pathways. And they show an even finer grain structures, early vision in blue, and then the ventral face pathway from posterior fusiform to mid fusiform to anterior fusiform is represented along this first dimension. And then the second dimension distinguishes areas associated with person knowledge from high-level face representations. So this is from the same movie data. Okay, this is not a face data set. This is the movie data set. And so you can see there's more information in the representational geometry if you look at even finer grained areal distinctions. This is just a paper that Andy Connolly published last year on representation of taxonomic continuum and a representation of threat or dangerousness of animals. And I can't go, this is complicated, but it shows a very nice progression from a visual representation to a representation of threat, or a visual representation to a representation of taxonomy as you go through pathways that's shown by changes in the representational geometry for these animal species that are either more threatening or less threatening, or have higher level animacy, more primate-like, or less lower level, like insects. So these correlations between subjects is reflecting divergent domains of information. So it's not just reflecting some global factor. And then this increase in between subject correlation is also shown in a study by Sam Nastase, who's in the back there, that he just published this year in Cerebral Cortex, where he used hyperalignment based on a different movie, a nature documentary, to align the representational geometries for videos of animals of different taxa performing different behaviors. And you can see there's a dramatic increase in the between subject similarity of representational geometry here too. So this is generally, so we've confirmed that this is working with this data set and others. Now, another nasty comment that slowed us down getting this thing published was that people said, you say fine scale structure, but we don't believe it. We don't believe you're aligning fine scale structure. We think this is, this can all be explained by coarser structure. So we had to go, we did a couple of analyses, and I'm going to show you just one here, where we calculated a point spread function, which is how similar is the response tuning function in one cortical locus with that same cortical locus in other subjects. How similar is it to the response tuning function for neighboring cortical loci in other subjects? Or cortical loci are separated by 2 steps, et cetera. So if we're aligning fine scale structure, we expect to get high correlations for the same cortical locus, but dropping off quickly as you move 3-6 millimeters away in cortex. So this is the slope of that kind of that point spread function for these different areas, but this is the average. So the point spread function for the anatomically aligned data is in red. And you can see the correlation of a locus with itself is about the same as the correlation of a locus with neighbors, or even 2 steps away. There's a little bit of a drop-off, but they're quite similar. After hyperalignment, the correlation of a locus with itself is much higher. That's what we showed before. And it's very different from the correlation of that locus with neighbors. So we are picking up a fine-scale structure at the level of a single voxel. So that's what we're discovering here. So how about, I promised that I would talk about connectivity hyperalignment. So as I said, we can think about the space, the common model space, as also containing the connectivity of each voxel with targets elsewhere in the brain. So we set this up to define multiple targets across the brain. We have, I think, over 3,000 targets. And we look at the connectivity vector for each voxel within a cortical field. That's shown here in the lateral occipital cortex. And we think about the pattern of connectivity to each target as a vector in this space. It can be a voxel space in the individual brain, and we're going to try to make a common model space of these connectivity vectors. So, so we look at the profile for one voxel, and we see whether or not the profile for one voxel, or for one dimension, is more highly correlated in the common model space than the connectivity profiles are for individual voxels in individual voxel space. So this middle row here shows the between-subject correlation of connectivity vectors, connectivity profiles, voxel by voxel. And you can see it's high. This is very consistent with other people's work. But after hyperalignment, these profiles are much more, yeah, Adina.
When you say connectivity, do you mean on the basis of TDI or just the...
Okay, this is functional connectivity. So what you look at is the correlation of the time series for a voxel with time series harvested from other parts of the brain. And we're doing this with resting state data. Chris?
So the targets here, right, for this to work, the targets need to be the same across subjects. So I guess is the idea that the M&I alignment is enough to align the targets because they're kind of close, but not enough to align the voxels?
Yeah, I've got a long answer for that. And that is that we define the targets as searchlights, okay, which takes care of some of the slop in terms of alignment. And then we do a PCA on the searchlight, and so that we don't have just a single time series from a searchlight, but we use the top 3 PCs. So we get a little bit more information from each one of those searchlights. And we have a very involved procedure for deriving PCs that are shared across brains. So you can see it's a the correlation of connectivity profiles is much higher after hyperalignment. We can also do this within subject, because within the HCP data, we can look at the correlation of the connectivity profile from one resting state session and another session collected in the same subject on a different day. Okay? So within subject, this is the kind of correlation of connectivity profiles that you get, which is much higher than the between subject correlation of connectivity profiles. So then we did a point spread function analysis of these connectivity profiles. How similar are they across subjects? And we showed that in the anatomically aligned data here, the connectivity profile for a cortical locus is pretty much the same as its neighbors or 2 steps away. So it's quite a flat point spread function. Which, but after we hyperaligned the data, now the connectivity profile for a cortical locus is much more similar to the same location than it is to its neighbors. Okay? Yeah.
So this is resting state data. So for Indiana Jones, it makes sense that brains would have similar patterns of activity, but why would you expect that in resting state?
These aren't patterns of activity now. These are patterns of connectivity. So for each location, you're looking at the correlation of the time series during the resting state for that subject with the time series in the resting state in other locations. Yeah.
Do we have voxel-to-voxel patterns of looking at correlation between time series, or are there multivariate ways of looking at how different voxels might correlate with each other, like canonical correlation or other methods that you could look at multiple time series at the same time?
Yeah. So you can, you can look at the similarity between 2 subjects or 2 brain areas as, using canonical correlation. We use the Procrusteus transformation between subjects because we're taking advantage of the shared representational geometry.
So it's no longer in voxel space,
it's in an information space.
information.
Yeah.
So it could be multivariate, actually.
Yeah. And the other interesting thing here is that the between subject correlation after hyperalignment of connectivity profiles is actually higher, significantly higher, not just a little bit, than the within subject correlation. And that's just a matter of how much data you have. Because clearly, the best estimate for one subject connectome should be that own subject's data. But when you're basing it on 15 minutes of data, it's a noisy estimate. Okay. So let's see, what am I going to go through here? I could talk about modeling topography as multiplex topographic functions. which works really nicely. Believe me.
Sorry, we can do 10 minutes for sure.
Well, I'll come back if, okay. So, so let me just summarize what this common model is, okay, because it's a complicated thing. And it has, the basic components are this high-dimensional information space for both representation and connectivity. Then transformation matrices that are specific to each subject. So the space is common across brains. Transformation matrices are specific that will project individual data into the common space or data from the common space back into individual brain spaces. And then there's the algorithm, which is hyperalignment, for calculating these transformation matrices. These dimensions, so what is the space? It consists of dimensions. So it's these columns in a data matrix that can be a response data matrix or a connectivity data matrix. And each one of those columns has a response tuning function, and the goal is to make those response tuning functions as common across brains as possible. And these, using this method, we can model population response that carry very fine distinctions, like the distinction between the response to monkeys or lemurs, or the distinction between two very similar time segments in a movie. And they capture variations with a fine spatial granularity down to a single voxel, and they're valid for diverse domains of information. This isn't just a visual model. It's really a model of representation in the whole brain. Each dimension also has a functional connectivity vector with other parts of the brain. And these functional connectivity vectors are common across brains. And they also capture variations with a finer spatial granularity than people had suspected before. Almost all previous work on the human connectome assumes that the response in a cortical area is homogeneous across the voxels. And we're showing that it's not as homogeneous as we thought. There's a fine-grained structure that should be exploited. And each one of these dimensions also is associated with a topographic basis function. That's the part that I just skimmed over. And these topographic basis functions are individual specific. They exist in the transformation matrix. And you can model an individual's functional topography as a superimposition or multiplexing of these topographic basis functions. And that actually works with high fidelity. So for example, this is an individual subject's retinotopy. So this is the polar angle map in V1, V2, V3. This map is data that have been projected from other subjects through the common model space into this subject's anatomy to map out an estimate of where the representation is of different polar angle locations based on other subject brain responses. And you can see it's strikingly similar to the subject's own retinotopy map. So functional anatomy is not lost in the common model, okay? So we have these topographic basis functions, that's what's shown down here, that let us model things like retinotopy or category selective areas, faces versus objects, et cetera. So it captures common bases for fine-grained structure, local variations of response tuning functions. So it's capturing both fine-grained structure and coarse-grained areal topography. Now finally, this is an important point. There is no canonical brain here. We don't have a group brain. Group brains don't exist, right? We each have our own individual brain. And so we're really modeling each individual brain with a set of basis functions that are shared across brains. So it's not a canonical brain. It's a common computational model. So how important is this fine scale structure in the brain? Well, it affords 7 times higher local between subject multivariate pattern classification, and greatly increases by nearly 2-fold the inter-subject correlations of response tuning profiles, local representational geometries, connectivity profiles. So really, this fine scale structure is a larger factor than the coarse areal structure. And you can think of it, I think of it as the dark matter of cortical functional architecture. It's carrying a lot more information about how the brain works. So let's talk a little bit about individual differences for 3 minutes, just so I can have a nod to Fei Long. Where's Fei Long here? There he is. There's Fei Long. This is his work in progress. So the goal of this model is to find what's common across brains. And after that, you might think that the residuals around this common model that are left in an individual subject's data is noise. In fact, we've been treating it as noise so far, right? And so it should be that the differences between brains are smaller, and that they're noisier, they're less meaningful. They're definitely smaller because the hyperalignment model works. So we have very smaller individual differences in functional response and functional connectivity across brains. So we asked, are those smaller differences noisy? Fei Long asked this. So the way he analyzes this is now we have a matrix of individual differences. So how similar is one subject to another in terms of their response to the movie, or how similar is one subject to another in terms of their functional connectivity for the whole brain. And you can see that, for example, subject 14 is pretty similar to everybody else, or the subject. And some subjects are very different, like subject 5 here, okay? So some subjects are similar to others, some have pairwise similarities. There's a structure in this. Now, so does the structure of individual differences, this individual difference space, is this less reliable after hyperalignment? So this is the reliability of this space for local fields. And you can see it's most reliable in the sensory perceptual areas, but still there's some reliability in prefrontal cortex and et cetera. After hyperalignment, we would expect this to go down because these the differences between brains are smaller. Instead, it goes way up. So somehow, by factoring out, by aligning the functional architecture, we're finding differences in how individual loci respond to a stimulus. The response tuning profiles are better aligned, the residuals of those response tuning profiles. This is surprising, and we're still thinking through what this means, but it looks like very powerful. And then do these differences mean anything? So Fei Long took data from the Human Connectome project, did a hyperalignment, or looked at individual differences in their connectivity profiles, and tried to correlate that with performance on a psychometric measure of verbal intelligence. And this is the map of correlations between connectivity patterns and picture vocabulary. So again, after hyperalignment, are these more reliable differences actually related to something like cognitive ability? And the correlations go way up. So these, whatever is left over, after we factor out these differences due to misaligned functional anatomy, gives us a better estimate of how subjects differ from each other. And I'm done. I thank you for your attention. Oh, I've got to show the last slide. This is Jarek when he first arrived at Dartmouth in 2008 with Michael Hanke. There's Baker Tower behind them. And you've been hearing a lot from Jarek. Too bad Michael can't come here and be part of this too. Here are some papers that we've got, one that we just published in Cerebral Cortex, one that's under review, but is available on bioRxiv by Gunta Pali. And then I hope most of you know about all these data sets that Michael Hanke is now making accessible. He's a big believer in open science, and he's producing these really large, detailed data sets that you can do anything you want to with. And you don't even have to, he doesn't expect to be an author on whatever you do. He likes to be acknowledged, of course. He wants the source to be acknowledged. But, so this is just a great source of data to try your ideas out on. Thanks for your attention. Bo.
That's great. What can we infer about differentially worse performance of hyperalignment in certain areas? So like what assumptions of the hyperalignment model are being violated in those areas? Is it, people are representing different things, or it's not a geometry, or what?
And so, you would, the areas that have worse alignment, the increment's about the same in terms of like a percent increment. but the alignment is worse in terms of intersubject correlation afterwards, and it was a lot worse before, probably because those areas like prefrontal areas are responding while watching a movie in a more idiosyncratic way, and that if the two subjects are having very different experiences, while they watch the movie, you're never going to align their response topographies. The basic assumption is that the responses are carrying the same information. And if they don't carry the same information, they're not going to align. Jeremy.
So would you advocate for kind of incorporating connectivity hyperalignment into like a standard pre-processing pipeline like motion correction or?
Yeah, I would. Yeah, I would. I think that, if, in fact, one of the...
Or I guess, like, would you see, like, are there any circumstances where that kind of wouldn't be a good idea, or, yeah?
I don't know. I mean, there probably are there are probably some pitfalls that we haven't discovered yet. But one of Swaroop's projects, I still have my hooks in him, okay? And he's developing a common connectome model based on 80 selected HCP subjects. Katerina's not here yet, right? Katerina Graton, she gave him the IDs for the 80 subjects in the HCP have the cleanest data. And he's building a common connectome, and he's going to build the tools so that people can take their own resting state data and hyperlink it to that common connectome. So the goal is that you won't have to derive your own common connectome. There'll be one common connectome that everybody can align their data to if they collect resting state data.
Standard anatomic.
Right. Yeah. But it's not available yet. So you can write to Swaroop and say, when's it going to be available?
Fine.
Yeah. I'm looking at you.
So this is a little bit of a follow-up on Bo's question. Many areas of the brain are very much engaged with what's happening right now in front of them, but some other areas are not at the same time course as the other ones.
Right.
However, when you're in a movie that is very engaging, most of the people might actually be flashbacking to another part, so other parts of the brain are integrating information or being reminded of it.
Yeah.
And it seems like with a little bit of asynchronous pattern matching for some higher brain areas, it might be that some of the regions that are not performing currently well might be able to perform.
Yeah, we haven't tried that yet. So you're saying that if we varied the timestamp in these areas, we might get better correlation because people are thinking about things at different. They're out of sync, but they're thinking about the same information.
So
Yeah.
it's in this context, but my PFC likes to go back to 20 minutes ago sometimes, and my parietal cortex to 5 minutes ago, and different
Right.
times.
That's a very good suggestion. And we know that the selection of the movie makes a difference too. So if you this is a pretty mindless movie. So you're not really analyzing the deep psychological states of these people.
We will be replaying so many other things.
Yeah. And Uri Hassan, who does, who works with the same kind of stimuli, but it tends to use a between subject correlation, voxel by voxel approach, although he's branching out now, has found that he gets more between subject correlation in areas like prefrontal cortex if it's something that makes people think about what are the people's motivations, or what are people thinking, what are their false beliefs. So people watch a really cool Alfred Hitchcock segment and gets much better between subject synchrony in medial prefrontal cortex. Yeah.
So you're applying this to fMRI data where you can get really fine-grained structure information. Would it be at all useful to use this in other modalities like EEG and fMRIs where the placement of the sources is external, and there's human error with exactly placing sources onto someone's head? In which case, maybe this would improve if you use this to align information that way. Would it be at all useful, or is it just not fine-grained enough that this really addresses that?
Well, if you use other kinds of data, like MEG data, it's much richer in terms of the temporal resolution. And so we've actually collected some MEG data sets and tried to do hyperalignment, and it kind of works, but there's clearly something that we aren't doing quite right yet. So so somehow you have to align things better in time, and this kind of idea of sliding the time window back and forth may be really much more critical for MEG data. Yeah, the hope is that we can use the fMRI data with the MEG data to kind of identify where the sources should be. We haven't made much progress yet, but it's a great idea. I would love to work on that or see somebody else working on it. Yeah.
So you described transforming data from common space back into particular subject space.
Right.
And so I'm wondering, and perhaps you covered it in this weekend, can you take a novel stimuli that's novel to subject B, not novel to subject A, and you can maybe get the signature in common space for that stimulus from subject A, and then be able to identify it reliably in subject B without even having calibrated on subject B.
Yeah, that works really well. So for example, we derived the model based on the movie data, and then with those transformation matrices, we can apply those to brain data from any experiment. And so we did that with the animal species experiment. Okay, so we took the data from that, and using the transformation matrices from the movie, we transformed these data into the common model space defined by the movie. So now we have pattern vectors for these animal species that don't exist in the movie. And it works beautifully. So we get between subject classification of the animal species that actually is a little bit better than within subject classification. So absolutely, that's one thing that works. Yeah.
You said that you allow for rotation, the Procrustes transform, or whatever it's called. How come you don't allow for different kinds of changes to data, like skewing it or stretching it?
Why rigid body?
Huh?
Why a rigid body and not something that can skew or shear?
Okay, so again, the representational geometry is a set of distances between response pattern vectors. And we know that's very consistent across subjects. So with a rigid transformation, it preserves that. So the distances between the vectors is just the same after rotation as before rotation. Now, this one method that the group at Princeton has worked on is a regularized CCA. And that allows a little bit of this kind of distortion you're talking about. And there's a parameter that you can set which makes it completely freeform to very highly constrained. When the very highly constrained CCA is Procrustes. It's the same thing. So you go from Procrustes to unconstrained. And they find that if you relax it a little bit, it seems to work a little bit better. So it's promising. We haven't been able to replicate it when we try it ourselves. So it's one of those things that it really needs to be developed, because it may be that a little bit of relaxation of that orthogonality constraint will make it more powerful.
So there's not some theoretical reason that you think the different brains would only be able to be rotated and not stretched and transformed in other ways?
Pardon me?
Like some theoretical reason, like this is how the brain works, or this is how different brains.
Yeah, I mean, we think representational geometry is actually an incredibly important insight. That the way we represent information, the reason that you and I can talk, is that we have similarities among the things we're talking about. I see red and pink as similar. You see red and pink as similar, right? And that it's this simple idea of things that are similar should be similar in different people's brains. Things that are dissimilar should be dissimilar. Is something that is kind of built into how our brains are built. Yeah.
So if you use Indiana Jones,
Hmm?
Indiana Jones is
Yeah.
awesome. But I could imagine that given what you learned, you might be able to design an optimum movie for hyperalignment or an optimum state.
Great question. So, as I said, this common information space is the same thing for response patterns and connectivity, and when we derive it on Indiana Jones, it transfers to representation of animals. It may be possible to get and we're estimating thousands of parameters in these transformation matrices, and so getting a more finely tuned estimation should make it more generally valid. And it may be a different movie, it may be more data. One thing we're doing is we have different movies. So we use David Attenborough Nature movie now for aligning representational spaces for knowledgeable animals. We don't even know if it works better yet. We just assume it does. Sam's going to answer that for us in a couple months. Mateo is building a different stimulus for aligning face representations, really hitting the subjects with lots and lots of face stimuli. So.
But why not, for example, give only visual stimuli, only auditory stimuli, and then something more complicated, and do separate alignment for different radios?
Well, yeah, that's the other possibility is that there's different common models optimized for different domains. Is there one common model that's going to work for everything, or do we have different ones for different, But in terms of efficiency, we're just finding that if we have people engaged in a really, dynamic, natural stimulus with lots of variety, we get a more general validity than if we have them engaged in like a simpler experiment. We've run the same analysis, model derivation based on experimental data where they just look at still images of a small number of categories. It works great for those categories. It doesn't generalize to other stuff at all. So.
In functional connectivity hyperalignment, you have dimensions that are a combination of different voxels. Do you look at the individual contribution of each voxel, and are they generally from the same region across subjects? That's my question.
So are the distributions for a voxel from the same region?
I mean the dimension in the context.
Oh, okay. That's something that I really skipped over was how we aggregate, how we get a whole brain transformation matrix. And we were worried about taking information from the temporal lobe and putting it into the parietal lobe or something like that. So that's why we do the first step in searchlights that restricts the resampling of information to other locations that are close by in terms of anatomical location in the reference subject's brain as compared to the new subject's brain. Okay? And then when we aggregate it, we're only resampling information into locations that coexist in the searchlight in the new subject and the reference subject's brain. But within the field being hyperaligned, that information can be resampled anywhere. So and that's a worry that we're struggling with, because if you have a searchlight that covers V1 and V2, it's going to take information from a certain retinotopic location in V1 and put it into V2 and vice versa. And that's not good. So we're trying to solve that. Yeah?
Just to follow up on that question, it's really interesting. Have you tried using hyperalignment to align shuffled movies where it would have the same few ports?
Yeah, we haven't done that.
Feedback might be different and might be useful to see, what at all is the contribution of having an interesting movie.
Yeah, we haven't done that. That's a really good idea, because huh? What's that?
Sounds like a hackathon project this week.
Great hackathon project. So you can get Uri's data, right? Because Uri has a lot of data where he's showing people movies with different levels of shuffling, and what that shuffling does is it really upsets the predictive coding efficiency. So that while you're watching a movie, you really know what's coming up, especially with Steven Spielberg. He wants you to know what's coming up, right? And that probably is a large part of what we're aligning is the prediction signal. That's why one reason I think this works well. And so I would predict that with shuffled movies, it's not going to work as well. That'd be really cool. Hackathon project. All right.
Let's thank Jim.